Hello, it's my pleasure to introduce Dr. Jim Kublin and Dr. Alexandra Schuetz. Jim is from, of course, the Executive Director of the HVTN, and Alexandra, the Chief of Cellular Immunology at MHRP. And they'll be talking to us about the mucosal immune activation from the female genital tract in 702 and RV144 via the RV306 trial. Go ahead, Jim.
Thanks, Nicole. And thanks to Alex as well for working with me on this slide deck and provide you a brief overview of our investigation into the potential risk factors of HIV acquisition in HIV efficacy trials, focusing again on HVTN702 and RV144, and in this case, an additional study RV306 that MHRP conducted. And so the hypothesis and question here really is, could a higher inflammatory vaginal environment, potentially driven by different microbial community types in South African women compared to Thai women, have contributed in part to the differential vaccine efficacy between these two clinical trials by increasing the risk of HIV infection among certain subgroups? So carrying on to some of Peggy's question, really a better understanding of population and or geographical differences that impact HIV acquisition when designing future efficacy trials may allow for a better stratification and selection of potential cohorts and aid in the interpretation of data, also building on the questions around the social behavioral data that's been collected from these clinical trials and this unique finding of segregation of risk and efficacy. So the background of this has really been evolving over the past decade, particularly in sub-Saharan women. They are at exceptionally high risk of becoming HIV infected, and the recent data presented and published around HVTN702 highlights that, as do many of the efficacy studies conducted in that region over the past decade. And more we've seen that vaginal inflammation, such as high levels of HIV target cells, recruiting chemokines, and genital inflammatory profiles increases HIV infection susceptibility. And then the vaginal microbiome and microbial function impacts the local inflammatory environment, with women who have non-lactobacillus dominant microbiota displaying higher inflammation and producing less lactic acid and demonstrating higher risk for HIV acquisition. And this is simply illustrated in the figure on the bottom right from a recent review. So we know that the microbiome in women in sub-Saharan Africa do display significant differences. They contain fewer lactobacillus species and more diverse communities, in contrast to what we typically think of as the gut microbiome diversity, when it's high, being very good. That increased diversity within the vaginal microbiome may put women at increased risk for sexually transmitted infections. So there's data comparing the vaginal microbiome based on geographical location, but these are scarce. However, multiple studies have demonstrated differences in the vaginal microbiome based on races and ethnicities. And then these lactobacillus dominant communities were found in 80% of Asian and 60% among Black women, with Black women having a higher prevalence of communities with greater diversity of taxa, and again, a predominance of these strict anaerobes illustrated in group 4 on the pie charts on the right. So the RV306 study design is illustrated here. It has the POX protein combination that was typical from the studies that Georgia had described. And they had additional late boosts at varying intervals, but group 2, that in which there's a boost at month 12, is that which closely reflects HVTN702. And then additionally, these other groups looked at AIDS vax only at month 12, and then again the POX protein combination at months 15 and 18. MHRP collected vaginal secretion by soft cup at these time points highlighted by the asterisks, which does nicely overlay with some of the mucosal sampling that we did in HVTN702. So in this group 2, specific characteristics are that there were 105 people enrolled, split between men and women, with fortunately a higher vaccine versus placebo. And that's relevant as we think about how to select samples from HVTN702 that we could match with this cohort. We have 38 of the 56 female participants consenting to consecutive collection of vaginal secretions. And these secretions were collected according to a specific protocol. We've seen plenty of this schema from HVTN702, but just as a reminder, the total sample size considerably larger. It was conducted across these clinical trial sites in South Africa. The HVTN702 mucosal sub-study, however, was specifically targeting the Isipingo and Verulam clinical trial sites in Durban. And the mucosal sampling schema is illustrated here. We had baseline samples that could be collected, and the modification of the sub-study was made so that these could be collected anytime within essentially the first 6 months of the enrollment visit. And then we did have an additional collection time point at the peak immunogenicity time point, or 6.5 months, and additionally at 12.5 months. For women, we had 2 vaginal swabs, 1 vaginal fluid soft cup, and 2 rectal swabs. And for men, we collected 2 rectal swabs and 2 penile swabs. And this is the data on the 702 mucosal sampling from Isipingo and Verulam. We had 370 men and women provide mucosal sampling. As you can see, split here between the 2 sites, and the number of HIV infections that occurred, again highlighting the significant burden of infection and disease among women. We did have a single case among the 89 men who participated in the study. And as a result of the tremendous effort by the clinical trial team, as you can see here, a very large number of the different sample types that were collected in the sub-study. So the proposed laboratory assessments are focused on defining the inflammatory vaginal environment and microbiome in women who were enrolled in these 2 clinical trials at baseline, 2 weeks after the RV144 series at 6.5 months, and then 2 weeks post the 12-month boost. For an investigation on the assessment of inflammatory vaginal environment using a combination of cytokine chemokine levels and proteomics, and assessing the vaginal microbiome by proteomics, including information about bacterial products, the metabolites, antimicrobial peptides, in order to define if different microbial communities might be driving different levels of inflammation in these different communities. A very unique investigation across these different geographic communities. So briefly, to thank the RV306 study participants, the tremendous effort by MHRP and all of their partners and funders. We also, as Georgia highlighted, have a tremendous team that supported HVTN702, with special thanks to Glenda and Nicole and Mary Allen for their input on the HVTN702 sub-study. All of the sites that participated in the HVTN702, again highlighting Isipingo and Verulam, who participated in the 702 sub-study. And a particular thanks to Geeta Ramji. Geeta and I worked most on the HVTN702 sub-study. She was tireless in her commitment to see the sub-study through and working with the South African MRC at these 2 sites. And I love this photo of her because it clearly she's clearly communicating to us that you could not pull the wool over her eyes. So with that, we'll open it up for questions.
I'm not seeing any questions posed yet. Well, there is one, I think it's already been answered about will we have information about mucosal cytokines between RV144 and 702, and Alex has responded already that that's part of the suggested proposal. The data is not available just yet, but we look forward to the interesting results coming out of this proposal from Jim and Alex.
Maybe I can ask a question. So Jim, so do you think that the correlate will be different depending on the level of inflammation? And is that separate, or is it linked to the behavioral aspects that we heard about in the last section?
Yeah, that's a great question. You know, and when we put the sub-study together, of course, we didn't know there wouldn't be vaccine efficacy, right? So we did include the opportunity to collect samples for BAMA antibodies at the mucosal surface. And we've never really investigated or had the opportunity to look at that in the context of a profound understanding of that local inflammatory milieu that's really established by the microbiome and its products. So that would be an opportunity to look at it.
And can you tell us, is there evidence from other vaccines that the mucosal, the microbiome makes an impact? Like, I'm just thinking about respiratory pathogens or, you know, HPV. I'm just thinking, like, what do you and Alex, and maybe I want to shoot this at Alex because I want to tease her a little bit, but what do you, you know, what do we know from other vaccines and the importance of microbiome and local inflammation and shifting how the vaccine mechanism might vary, and why that might be so important, and trying to resolve why RV144 might be completely different than 702, not just based on the level of immunity that's induced?
I mean, there are studies out there that the gut microbiome is significantly influencing the response to vaccines. And there is a recent study, I think they took a quadrivalent influenza vaccine and depleted the gut microbiome by antibiotics, and found out and had that as one group, and then the control group did not receive any antibiotics. And it turned out that in the group that received the antibiotics, you saw a significantly lower influenza-specific antibody response. And it's due, I think, or they think, or it's speculated that it's due to the ongoing inflammation that is caused to the microbiome. So again, that's gut, and as Jim has pointed out, gut is very different, or a healthy gut microbiome looks very different from a healthy vaginal microbiome, but you could imagine similar effects because it could have a more systemic impact if your microbiome is disrupted.
Is there any data obtained so far, Jim or others, that suggest that there wasn't true randomization in 702?
That there was not true randomization?
Yeah.
Not yet.
Because I think at the end of the day, we're going to need a vaccine that's going to work in everyone, not just some people that have certain mucosal environments or behaviorals or whatever. So is there any data so far that there are differences in the controls and vaccines in any aspect that you've measured so far? Or did randomization actually work?
Randomization worked as far as we know. You know, again, we haven't interrogated the local milieu within the mucosal surfaces to confirm that. And I agree, we certainly don't want to have an indication for those with a high lactobacillus vaginal microbiome. That said, if there's an opportunity, a proof of concept that could be demonstrated within those who, if, as Galit's question, were extended and confirmed by an investigation that correlates of protection were within one of these subgroups, how then would you induce or increase those correlates across all different populations? Of course, that would be the extension of that.